So it's about time. So 8 p.m. We'll start at 8 p.m. Indian Standard Time. So I think we can start now. So hello everyone, and thank you again for joining this webinar on high-throughput biomedical data analysis. This webinar is organized by our team here at Pine Biotech. The webinar will cover several topics from the upcoming Biomedical Data Science program that we will be launching on September 16th. So at Pine Biotech, we are constantly working towards creating meaningful curriculums in advanced topics in bioinformatics and data science to people with or without technical or engineering backgrounds. So in our OmicsLogic courses, our focus is to share the logic of the analysis and also to apply these methods in competitive projects and the projects that actually stands out in cutting edge bioinformatics and biomedical research. So in this program, we will focus, more on the data science techniques that has to be embedded in our analysis so that in future research, you can move into the right direction and get the maximum insights from your data. So the goal of today's webinar is today we will be introducing biomedical data analysis techniques to you and approaches that you can master and adopt to help conduct better research, of course, and learn about how data science is changing, the entire spectrum of medical research. So from diagnostics to treatment selection and development of new therapeutics and drugs, it's very important the role that data science is playing these days because of the enormous volume of data that has been generated on a regular basis, now on an everyday basis. So during this presentation, we will walk you through the smart syllabus of this program. This unique data science online program is designed in a way so that everyone who joins will have a complete data science research experience working on biomedical data sets. We have 4 aspects. So we have 4 different aspects of this program. So we will start with the understanding of biomedical challenges, and then we will move into the exploratory data analysis, and that would be followed by data analysis and visualization. You can see here, we are trying to build better visualization for the data sets that we are obtaining and that we are processing and that we are trying to separate or classify. So after the data visualization, we will look into the machine learning techniques, how these machine learning techniques are being helpful in understanding the rightful, the meaning out of these big data sets. So the course, as it has been already talked about in the previous webinar, so the course will start from a low introductory level. So we will start from the very basics, and then we will gradually together, we will advance towards the various techniques used to understand this high dimensional data. So the biomedical data challenge that we will be addressing and really focusing on is the molecular data, right? The multi-omics data that we have already discussed in the previous webinar. But here again, the multi-omics data, we will be working towards the goal of, precision medicine. That is something that we can look forward to, and that has been in practice already in many places using this enormous and meaningful data that we have been generating from even like from a patient sample and different environmental factors. There are so many things. So the biomedical challenge that will be addressed in this journey. So you will see, we will see how effectively we can utilize these tools. So one of such tool is our TBioInfo platform. We will be using TBioInfo platform, which is a seamless and a very intuitive and AI-guided platform. We'll be using that for our analysis of high-throughput data. Followed by that, we'll be using a lot of MS Excel and RStudio, where we can take the output of the program and learn how to visualize it better and understand the real meaning from those data sets. So here we are trying to walk you through this journey of data science, where we will be talking about the challenges. We'll start from the challenges, and then we will move into the exploratory data analysis. We will look into the data visualization, and then we will understand what does this means. So, I mean, we will be guided throughout this journey. This program is organized by a skilled team of experts that have a diverse perspective on challenges and methods that we will be covering. So we have Darko Medin, who is a data scientist at the University of Montenegro. He's working on a biomedical and pharmaceutical clinical studies. And myself, Mohit Mazumdar, I have a PhD in computational biology and machine learning. So before joining, so can I ask you to please mute yourself so that we can have a seamless lecture. So yeah, in this course, we'll be sharing valuable information about our experience working with research labs and various healthcare industry. So before joining the academia, I have worked on high-end bioinformatics projects and problems well defined by pharmaceutical companies working towards drug discovery, patient treatment, or personalized medicine. So we'll be sharing all those insights that we have gathered throughout our time, and we will use that experience to guide you better during this journey. So, I mean, going into the details, as I said, we will be discussing the syllabus here. We will be discussing what we will, what we are going to, cover during this whole process. So now let's move on to the details of each session. So let's look what we are going to cover day by day. So after the introductory session on 16th of September, so we start on the first day, 16th of September, where we'll be introducing everyone to the platform and what all courses, what all material is available for you to take and learn. So after that session, we will be talking about various data types in high throughput data analysis. So last time in the previous webinar, we discussed about the raw data like FASTQ files, SRF files, etc. But the range of available resources goes wider than that. Upon directional search, not only that you can find raw data, but you will also see the lot of processed data. When you go to repositories like TCGA and the repositories like geo data sets, where we are finding a lot of large volumes of data deposited on a regular basis, you will also find table of expression. So the table expression means that the data has been already processed from raw to now you have a table of expression. You will find VCF files that contains the mutational variants. You will directly find those files, and you can also find 16 as ribosome O2Us. So those things you will get for metagenomics analysis. So those things are also available for you to learn and explore the data that is available out there. So in this session, we will learn about the kind of data that I just discussed. In this session, we will also understand what can we do with this data and how do we manage, how do we analyze this using the intuitive AI-guided T-Bioinfo platform. So yes, last time we showed you a demonstration of how easily you can access the platform. The participants of this program, which will start on, as I said, which will start on 16th of September, will have access to our analytical T-Bioinfo platform. And the educational platform has all the resources, plus the educational platform, which has all the resources associated with the course, and the material available with this program will be communicated to you. So after that, we will learn about the various data types associated with different analysis. The platform offers meaningful analysis and methodologies for various multi-omics data analysis and interpretation. So last time we looked into RNA-seq analysis. In this session, during the course, we will go deep into the processing of NGS data. We will look into the cleaning of data. We will look into the processing of data using our semantic pipelines. So this is one of the biggest USPs. So using the different sections of this analytical platform. So we have different sections, as last time you saw, there were like sections for NGS data analysis, there's section for system biology analysis, there are sections for machine learning analysis, data mining, to be very precise. So using these different sections, the platform which enables us to, easily, very easily upload data and also integrate the raw data following by the, processes like cleaning, processing the data using the logical schema of algorithms. So after the analysis, what we are generating is a table of expression from a raw FASTQ file, right? So this is a specialized bioinformatics pipeline, as it is shown here. They are connected to a logical scheme of analysis, and the same analysis like this pipeline is, as you can see here. What is the advantage of learning about the pipelines which are hosted in T-Bioinfo platform? There's a question like this that comes up. So the answer to it is like the same analysis could be implemented not only here, but outside the platform as well. So what you want to learn, what would make it happen? So you have to follow the same logic, right? Once you understand the logic of how to connect and how to, why to connect these different steps, and if you understand that, so you can translate that into, any kind of environment. And that is one of the benefits of this program. So yeah, if I have to summarize here, we will start with the introduction, and then we will look into the big data coming from these high throughput techniques. For example, here, we will extract the table of expression, as I said, and then we will move into the statistical analysis. So for the next section, I would like to invite Darko Medin to talk about the statistical properties of the data. So I'll just stop sharing, and I will let Darko to share his screen and give you the insights.
Thank you, Mohit. So, as Mohit said, sometimes we will deal with huge amounts of data. Now, this data will come in a variety of formats. So, for example, our data could come in a table format. We will want to summarize our data, for example, find central points of the data and explain the variability inside it. From the descriptive statistics, we will get an output that contains numerical values like means or medians, or for example, standard deviations. In this case, we are showing you an example of visualizing the medians on the left side of the image. Take a look at the medians and take a look at how they relate to the whole data. We'll deal with many other aspects of descriptive statistics analysis. For example, here you can see Q1, Q3 interquartile range and many other statistics. We will cover them in detail during the program. On the other hand, data could come on a scales of different magnitude. So these magnitudes should be aligned in order for us to create a more comparable data. We will use a method called quantile normalization, which can be accessed on the T-Bioinfo platform. And we will be aligning these medians from different scales, different file formats, different data formats. And once we align them, they will be more comparable. Later in the course, we'll be dealing with more data visualization tools for exploratory analysis. We'll be using box plots, histograms, scatter plots to analyze correlations and associations between the variables and analyze the distribution of the data. This example. On the left, you can see two scatter plots explaining correlations between two variables or two parts of the data. For example, these could be two genes, and we can see the red line showing a trend of correlation between them. Now we can use these trend lines to analyze the nature of the association. But take a look at the upper scatter plot. Our data is agglomerated on the lower left part of the chart, and it's very hard to visualize the whole thing. But after normalizing the data, which is in this case non-normalized, typically RNA-seq data will be like this. But we could use we can also create a predictive model out of this, so we could predict values of y based on values of x. The whole point is to find a predictive model that is most accurate, that has least errors in it. So we'll be dealing with least error prediction models. On the other hand, we don't have to analyze two variables. We could include more variables in the analysis, and we could combine the regression with factor analysis to identify, for example, genes that could be important factors for certain disease. Now, when dealing with big data, very often we will have huge amount of variables, huge amount of data. For example, huge amounts of samples and huge amounts of genes in our data sets. We'll be dealing with data sets that contain several thousand genes. So we will want to choose the genes or parts of the data that will be most relevant to our research. For example, a large number of these genes are irrelevant for breast cancer research. Let's say we have several types of breast cancer samples, right? Now, large part of the genes will be normally expressed in them. Just a portion of the data will be important for us to make a distinction between these breast cancer subtypes. Use a method called PCA, or Principal Component Analysis, to extract parts of the data that are most relevant to our research. PCA does is it selects part of the data, let's say from a huge data set, that has the most variance in it. Why is this done? Because data that has more variance separates subgroups much better. So, Principal Component Analysis will create PCs, or principal components, in a certain order. For example, PC1, PC2, PC3, etc. And the first PCs will be the PCs that explain most of the variance, that are best used to separate our data. In this case, we have an example of breast cancer subtypes. For example, luminal subtype, which is marked as blue here, is separated on the PCA from the rest of the breast cancer subtypes by PC1, or the principal component that has most of the variance. The principal component analysis can be improved even more using software like R. So we can combine T-bioinfo platform to conduct initial PCA analysis and then combine this with tool like R or RStudio to improve our PCA. You can see on the right PCA that we actually separated 4 breast cancer subtypes, and we were able to customize the plots to draw some borders. We can review the overlaps between these subtypes and calculate their distances. We'll talk about this more in detail during the program. Now I will give the word back to Mohit on the clustering part.
Thank you, Darko. I mean, yes, I mean, you guys learned that unsupervised method is PCA is one of the way to look into the data, visualize it. So it's an unsupervised method, which means that the data you are looking at is not labeled, right? So if you guys can see my screen, can you guys see my screen?
Yes, we can see.
Thank you.
No.
You can't? I think it's just going to load in a while.
I can see it now.
Yeah, thank you. So yeah, thank you, Darko. I mean, so after performing the PCA, which helped us to visualize the data, so we are going into a flow here. First, we did what we did is we extracted data from the high throughput analysis from raw data. We came to expression table, and that expression table is nothing but something which is made of columns and rows. So we have a matrix of data which contains multiple information about different samples and different genes. So there are thousands of samples, there could be thousands of genes, right? So what PCA did was PCA helped us to visualize that data and that high dimensional data. So, okay, and then you saw that we can classify them into different groups using, our specialized customizations that are available within the platform. And also we used R especially to for a better visualization, and also that would be covered in much detail in the course. So we have a specialized course for R that helps people and that helps our participants to not only see the classification, but also derive meaningful biological interpretation from that data, so which is included in this course, of course. So we discussed about the PCA. Now what we will try, which is like another very popular unsupervised method, is clustering, right? So during this program we will use hands-on example with clustering algorithms. Like I think many of you have heard about the k-means clustering algorithm. We will cover k-means. We will do hierarchical clustering. We will do db-scan, which is mostly density based clustering, and we will combine them with heat maps to improve the visualization of the analysis, as I said. And we will find patterns in the signatures of those separate clusters. So in this session, during this session, we will learn about various clustering methods, algorithms that takes distances and boundaries to separate data. So yeah, one of the examples, like we will next we will go into one of the examples that are present in that we did in one of our breast cancer sample data set that we have been talking about. So this is just an overview of the supervised analysis methods that are available in the platform. So we have different methods, and those methods have different parameters. So hierarchical clustering orders the rows and the columns based on similarity. So this makes it easy to see correlation. Out of the results from hierarchical clustering, you would be able to see better correlations. So during the clustering session, we will use various examples. Like in this case, what you can see here is a section where you can see you can define parameters like linkage, distance, and the number of clusters that you expect to get from this data, transposition to understand to transpose between the rows and the column. So you will have different parameters that will be taught, and we will make you understand why this lineage method is better for a certain data set, why this distance is taken into consideration when we look into different data sets. So it's mostly about the application, not just the methodology. We will go into the algorithm in detail. We will talk about the application of that algorithm in a real data set. So I mean, what we are trying to do here, I mean, what is important in hierarchical clustering here is that the output of the hierarchical clustering gives us the separation, right? So it gives us a separation between different groups present in our unlabeled data set. The important question, however, is when we are doing hierarchical clustering, how do we determine the similarity of the sample, right? How do we determine which samples are similar to each other? Or in other words, how do we determine the distance between the data points, which are there, and then link them to different clusters that are present? So what we are looking at is, we are looking at the relationship between the samples based on their gene expression. And hierarchical clustering is based on a bottom-up approach. So we have classified the samples into separate groups using in the next step. So you can see here, like in the dendrogram here, which is shown. Excuse me. So yeah, I think, yeah, I got the mouse cursor. So the dendrogram, if you can see here, I mean, it divides the classes. If I draw a line here, it would divide the clusters into 4 classes: 1 here, 2 here, 3 here, 4 here. So you can see we can divide this data into 4 distinct classes, right? So using in the next step, I mean, once we have data, once we have classified samples into different groups, what could be the next step? So using the next step would be to use the supervised machine learning methodologies. So we have seen so far that we can possibly, group unknown, unlabeled data into, different clusters. So what could be the next step? The next step would be to separate these groups into different classes and then make a training and test data set to see how does this unsupervised learning algorithms that we would see how this unsupervised learning algorithms will work on the labeled data and how they perform. What we are trying to do here is to classify or find important genes that can, classify the breast cancer subtypes into different categories. So that's the purpose. That's the biological question that we are asking here. And we are using different algorithms which are giving us different outputs, which gives us different insights that why this gene is important, for, discriminating between maybe basal-like or Claudin-low or normal-like or, different cancer subtypes. So that's the purpose, and that's what we are going to learn in this course. So, I mean, we talked about unsupervised method. We talked about PCA. We talked about hierarchical clustering and different clustering methods. Now, as you saw, like here, you saw that we have been able to divide, different groups. You saw here in PCA, we have divided them into different groups. So we have now defined clusters. So the clusters are well separated so that we can make them into, into a training and test data set. So next is something called the supervised machine learning method. Now we have a training and a test data set to evaluate the performance of our classifier. So why we are using, so many different machine learning algorithms? So many machine learning algorithms you saw, like in one section, there are multiple machine learning methods available in the platform. So why we are using those machine learning methods, different machine learning methods on different data sets, different machine learning methods on a single data set? What insights does these different methods give us? So I will come to that in this webinar, but we will discuss it more and more in our in the program itself. So I mean, we saw how these trees are classifying data. So basically, if we have to separate everything into series of rules, so a decision tree is what? A decision tree asks the question yes or no. It starts with a random initialization. So we'll look at an example. I mean, then I think we will be able to understand better. We'll look into one example of breast cancer cell lines, and maybe then we'll be able to understand how actually decision tree works. So we are looking at a decision tree of 15 genes. So 15 genes plus those 15 genes are, we are looking at of the expression of those 15 genes from different samples. So we have different number of samples available. So we have a lot of basal sample available, a lot of Claudin low available, a lot of luminal available, and a lot of normal like samples available. So based on that data set, we are trying to classify them into something called this. So let me explain to you here what we are actually looking at. So when we are talking about differentially expressed genes, typically we refer to some sort of profile that looks like this. This is a gene, okay? So what we are looking at is here you can see a group of samples where the expression of the gene is upregulated. Here you see this gene is upregulated in a group of sample. So I think the fonts are very small, but the thing that I'm trying to tell you here is like these samples have been put into one specific, they have a high expression. It's quite visible here. This gene which is the output of the decision tree gives us uh, uh, gives us a separation based on single gene, that okay, if we choose this gene can help us separate this group into different sets. So in this example there are two genes. So there is one gene here, there is one gene here. So two genes out of 15 that are helping us classify these cell types, these subtypes into three groups. So gene 6299 here and gene 9888 here in combination can distinguish whether a sample belongs to cell type basal, luminal, and Claudin low. So how did we manage to understand this? How did we reach here? Those are the things that we will be covering in the in the program, that how we interpret a decision tree, how that algorithm is being able to come to a decision where it can use one gene or two gene to classify two big data sets. That is something that is decision tree, right? But there are many problems with the decision tree. As I said, there are several problems, one of which is that the algorithm works with random initialization. So from your data set, it will just start randomly from anywhere. That's one problem. The first tree will start from some place, and we will try to find the optimal genes that are separating the data sets into whatever classes that you saw. So the issue is in with the decision tree is the accuracy and the stability. So because you're doing a random initialization and you have generated one tree, and this is not highly accurate practice. So we have found a problem to this. We have found a problem of resolving this decision tree, and that solution is nothing else but another very popular method, I think, which is random forest, right? So random forest, what is happening in random forest, and why random forest of decision trees? So we are running actually multiple decision trees at a single time. That is called random forest. So when in random forest, instead of running one decision tree, we are running it many times, like, right? We have one instances. So the whole idea is to actually develop an algorithm that does actually run the decision tree many times in order to focus on the stability and the accuracy of the result, which was missing in the decision tree. So you see, right, here we have an instance. Okay, we started from an instance and there are multiple random initialization. So we are generating many trees, and then we see some predictions happening here, some predictions happening here. So this optimal path which led to this result could happen again. So it's not fixed that the initialization if already happened with one integer, it will not happen with the same integer again. So it can happen again and again. So what is the result? I mean after you doing it many times, you might ask that how do you conclude that what is the right optimal path or the right optimal genes, because we are getting so many instances. So what actually happens in random forest is, it's very interesting. It's like we do voting. So if this result is coming many times, if this result is coming many times, if this result is coming many times over and over, then the probability of that result to be correct is high, right? So that's how random forest work. We will go into much more detail where we will talk about, the algorithms, the booting and bootstrapping, all those things we'll cover, and we'll make you understand how it is, affecting the analysis of the data that we are doing, how, running different, this is a computationally expensive process because you're running so many different trees at the same time. So we'll discuss all those things in details, but this is an overview how we are going to deal with this data, and how, with relevant examples, we will be talking about this in detail. So yeah, so another example, like as you said, we saw the trees, right? We saw how trees and how they work. We saw the tree-based classification. Now we'll move into a line-based or a data transformation method, which is the LDA. This is also again a very popular method: discriminant analysis and classification. So what we are here trying to do is, like, the actual classification. So we are dividing the training data set and the test data set, and then we are looking at performance, how these separations are actually working using these different techniques. So one thing I talked about previously, that we are using different algorithms. We are using different algorithms on a single data set. How does that benefit us? So here you saw, I mean, here in decision tree, you got what? You got two genes, right? Here and here to classify. Here, actually, when you see the result of decision tree, you get a bunch of genes, not one gene, like maybe 15 gene, maybe 7 gene, maybe 8 gene, that is helping you to classify, or to segregate into different classes, or to tell that this is the gene that belongs to this class, something like that. So here you're getting a signature, you're getting a gene signature, because you're not getting one, two, but you're getting many. So you understood why we are using different methods, because different methods are not only giving us different results, but it's also giving us different insights from this data. So one data set which is that big have so much information. So different algorithms work in a different way to extract those information, which is equally important for us, for to conduct a good quality research. Even to many of you might work in industry where the demand is to reach to a conclusion which is more effective. So the data driven approaches, the hypothesis, before this there was a hypothesis driven approaches, now it's more data driven approaches. So what is the big benefit of data driven approach is that we are coming to a highly accurate answer, which helps us to reduce the bench work, or which is helping us to reduce the amount of the hard work, the amount of money that goes into bench, that amount of money that goes into doing experiments, wet lab experiments. So if we can have a very specific answer, we can just design an experiment for that, and that would help us not only to reduce the amount of expenditure, but the biggest advantage, one of the biggest advantages, the time. So in pharma, as we all have heard, uh, to reach out or to make a drug which is very effective, it takes like a huge amount of time. It's a 15-year or a long time scale, and also it has a lot of uh, problems in terms of uh, coming to a definitive result. So that those are the things that can be overcome while you use multiple methods like LDA, decision tree, and look into your data very closely, and then come to then come to a solution. So yeah, we are talking about discrimination analysis. To discriminant analysis, the idea is here. The idea here is to define, or discriminate between different types of samples. So we are removing those features which are not important. We are removing in an iterative process, and we are moving forward like this. So I mean, let's go into a little bit more detail into LDA. So in some sense, LDA, or linear discriminant analysis, which could be said as a supervised alternative of PCA. So PCA is unsupervised method, but LDA is something you can compare it with LDA. And how and why? Because instead of finding the best principal component that separates all the samples based on variance, here we will find some line that accurately separate our classes. And there are many different variations of LDA. Okay, so here you can see linear LDA, you can see quadratic LDA, you can see diagonal LDA. So these all are based on different assumptions about the distribution of the data. We will go into much detail in our class about the LDA process, how we are using different methods, and what are the different data dimensions that we are talking about. So another way, like LDA, we are doing it for one instance. An iterative version of LDA, just like we have an iterative version of decision tree, is called stepwise LDA. It is useful for the same sense as the random forest is useful. In comparison to decision tree, if we will select genes as the separation, different here we will get a best separation in a different principle or a different algorithm. The algorithm that LDA uses is called the greedy algorithm. So we will go into detail now, but go into such detail. The separation methods like LDA and SVM are something which helps us to understand the classification in a better manner, because we are training the data set, and then we are asking the classifier to classify new data sets which we have no clue about. So once you have developed this protocol, once you have developed a model, it's fairly easy in the next step to predict for the new classes. But the challenge remains in developing a good model. That is something that we want to discuss in this entire program, so that the output of the analysis that you're getting, many people use machine learning nowadays because it's very popular. What they don't understand is that how these different techniques deal data differently. So that is very important, I think, to move forward. So the result of stepwise LDA is going to be that. So we can get a good separation. We can understand based on which genes we selected and features, and then we can do downstream analysis of those genes and understand how the biology of it, how the interpretation of these genes correlate with our findings. So another method which I said is SVM, which is another very popular method. So I'll not go into much detail. So SVM is a very effective algorithm, which what it does is it takes the input space and it converts it into a new type of space. So where it is easy to separate your samples, right? So you can see here the samples are inside, are surrounded. So this is class 1, and the samples outside is class 2. So this is a difficult separation, So what we need to do is maybe transform this space. So how do we transform this space? This transform the trick here is kernel. SVM uses different type of kernels to transform this space so that it could improve the classification. There are radial basis kernels, there are linear kernels, there are different type of kernels that works and that helps us easy to transform and separate these spaces into different categories. So it's very fascinating. I mean, once you start applying this data and see different results, you'll understand how these methods actually work in tandem, or how these methods are working in a collaboration for a data set. So I mean, once you learn in this phase, we have initially learned about the data. Now we have learned about the techniques. Now in the next phase, we will move into the case studies, where we will see the application of both in one place. So I will just give the stage to Darko to tell more about the application of these techniques, the case studies that we will be covering in our in our upcoming program. Thank you. All to you, Darko, please. I'll just stop the sharing.
Okay. So, as you saw, we will be dealing with a variety of aspects from data science. We'll be dealing with huge data sets, mainstream statistics, machine learning algorithms, and many other things. But you don't need to have previous knowledge or experience regarding these matters, because we will be starting from the very basic things. Then we will go through all of these aspects we spoke about. For example, we will teach you some principles in how to choose which analysis or which algorithm to use for which data. For example, we have unlabeled data. We have not predefined the groups, and we will use unsupervised learning methods like PCA or age clustering. We have predefined the groups, and now we will use random forests and QDA or discriminant analysis to classify the data for us. We'll apply all these on hands-on exemplar projects. So on October the 21, we will start with projects from oncology areas. Now, in these projects, we will have various types of data. We will have data that are just images or visual data types. As you can see here, we have an X-ray, which is actually a mammogram, showing tumor on the picture. So they're used by doctors to identify early signs of cancers using X-ray. On the other hand, we use many other visual data types like biopsies, histologies, to analyze the visual aspect of the clinical data. Molecular data will complement this analysis. So, for example, we have biomarkers for some cancers, but it's just one biomarker, it's just one protein. What if there are many other processes that occur in the development of this cancer that we're not dealing with, right? So we want to analyze multiple types of data, for example, gene expression patterns and other molecular types of data, like the ones we see on the right. We can see a heat map here representing gene expressions. Green is a higher expression and red is lower expression. So we can see that there are more cancer subtypes here. Each one of them has its own pattern of gene expression. When we go to the project itself, modeling precision treatment of breast cancer, we will be dealing not just with molecular profiling and clinical data types, but also responses from 138 therapies. So these combined, and with the use of machine learning methods we mentioned, a trained model can be created that would be able to predict therapy response of each patient based on all these types of data. So in the end, a treatment response prediction model is created for 22 therapies, and each new patient that is analyzed based on these molecular profilings and breast cancer clinical data types, each treatment response could be predicted. 24th, we will start exemplar projects in microbiology area. So we will analyze Bacillus tuberculosis and drug resistance that is developed to antibiotics, but through omics data and using these data science methods. On the other hand, we will have a project that is related to Ebola or Ebola outbreaks in West Africa. This is an outbreak that occurred between 2013 and 2016 in various parts of West Africa, like Sierra Leone, Liberia, and many other. So the outbreak was very infective, so the virus itself became more infective because some changes occurred in the glycoproteins on the surface of the virus. How the virus infects human cells, it infects them by these glycoproteins. So human dendritic cells interact with the glycoproteins on the virus, and that's how the infection occurs. Now, a single change in these glycoproteins could make the virus more infective and spread much faster, like the situation we have at hand. So We will have an exemplar project with data sets from different parts, originating from different parts of Africa, containing multiple omics types, and we will use machine learning algorithms and data science to analyze. We'll create pipelines on the T-Bioinfo platform. We will map the transcriptions. We will use variant calling, differential variant calling, and we'll cover this in detail during the program. And in the end, we will identify these changes or mutations that cause the virus to become so infected. On the other hand, we will have projects from various areas. They're not limited just to oncology or microbiology. We will be analyzing exemplar projects from asthma studies, for example, or studies related to endocrinology, to diabetes. So that is the key concept of this program. We'll teach you how to analyze the data, how to access the data. We'll teach you how to perform data science and machine learning, but we will apply these to a large number of exemplar projects. So this is not just some online course. This is a hands-on, practically oriented program, and we will be there with you from the beginning to the end and implementation of these methods to answer questions regarding the program. We'll teach you how to access the data in a very easy way from sources like NCBI or Firebrowse or EMBL, Entrez, Uniprot, and many others. We will teach you how to interpret and download these data, a plan of analysis when you access the data. So this will be a 3-month, more than 3 months in duration. So in the first 2 months, we will be teaching you how to apply these methods. Then after October 31st, you will start with your own data process. You will select your own data sets, and you will have approximately more than a month to be guided by us with all arising issues in applying the methods we taught you before. So right now, I will give you some more insights into the program.
Thank you so much, Darko, and that was an excellent guidance that you gave us. So yeah, I mean, as you saw, I mean, we will be covering the theoretical part, and we will be talking about lots of hands-on practice during the whole process, and we will finish the details, and we'll be having one-on-one talk with you. We will be having group talks to discuss more about the details of these these projects that we'll be doing next. And after the projects, we'll be talking about the outcome of the project. So it's a journey, I think, which will be a very fascinating journey together, where we will collaborate and learn together on different domains. So we will start this on September the 16th, this month only. We only have 6 days to go for this program. And as you see, we are all very excited to start this program with a group of enthusiasts who wants to learn, and not only learn, but also apply these methods into real life problems. So what is this project, or this whole program is bringing, as we keep on saying, that we will go through the methods. We'll not just go through the methods and learn and understand about the details of each of these methods. We will also apply these methods to clinical research and pharmaceutical projects. Then we will design and prepare, you will design and prepare and plan your own analysis, which could be impactful. So based on these projects and analysis examples, what you will do is that you will learn how to handle important project an important biomedical research that could eventually lead to something translational, like a publication or like a grant, or different things. When you're working in a company, you're looking at a problem, and in the end you will obtain a certificate, an international certificate, which could be of high value, where you will be getting the knowledge which will be transfers into all these time, and we will be finishing this course at this point of time. So it does gives a lot of value. So we will start from 16th of September, and we will go till end of October during this journey, where we will be talking about, as you saw, we will be talking about the challenge first, and then we will look into the data and how the challenge is, and we have to understand that why this is a challenge, first of all, why this is such an issue, and what this platform brings you to simplify things and dive into the actual problem, rather than focusing on the methods, rather than focusing on the understanding the output of those methods. We will dive into something really translational that would help us to do something which is the need of the hour. So we will cover all these during our sessions, and it will be it will be always like this, will be an interactive session, where we will be connected to you using diff platforms that we have. We have various platforms on social media. We have WhatsApp groups. We have been reaching out to all of our students for different type of queries that they have. So be interactive, and it will be a collaborative effort between you and the Pine Biotech team and us. It's a journey that we're looking forward to. So and I think have any questions for us, during this presentation. So please go ahead, unmute yourself, or write in the chat and ask a question that if you have any questions regarding to the webinar and also the program.
Hello.
Hello.
Yeah.
Hello.
Hello.
Yeah. Hello.
Can you hear us?
Yeah. We can. I hear you. Yeah. We can. Yeah. We can. Yeah. Please go ahead.
Hello.
Yeah, Vamshi. Please go ahead, Vamshi.
Oh, yes. Yeah. You recognize me. Thank you for recognition.
Yeah. Please.
I just have this doubt. We use RStudio here, right?
Yeah.
So is it in the cloud computing, or we need to have the software on our own machines, or just like that?
So if I can answer that question. So RStudio is we will be using RStudio for better visualization.
Yes.
So we get the data from the cloud-based our server, and then we will look into manual installation of those programs in your computer. So we'll be guiding you how to do that as well. We'll be guiding you which libraries we will be using. So yeah. So there will be no issues, it's a downstream analysis. So that can be, different users have different, different publications, different user have different, need. So it's something which is personalized, but we'll be guiding you through the whole process.
Yeah, we will be guiding you from the basic things, like how to download the RStudio, and then how to use the platform or the Excel, or how to customize plots in the RStudio. So we'll be covering all those topics from the very beginning, and then going to more advanced things.
Great, thanks.
Yeah.
Okay.
Hello.
Hi, Sunita. Yeah.
Hi. Like for some time I have been facing this, and I have a question, like to access data to get data into the platform and work is being a little difficult for me, probably because of the size of the data. Is there like I probably have an extension to the same question that Vamsi asked just now. Is it possible actually to download these to have these tools working on Mac system and work on it, or do you think that this platform would help me for complete analysis that I want to do?
So we will be combining the platform with other tools, right? So where it's easier to use the platform, you will use the platform. But then you can combine these with tools like Excel, RStudio, and many other tools to overcome these obstacles, right? So you will do the data processing very quickly on the platform, but then you can customize it more in other tools.
Yeah,
And,
and the tools have been tested in different platforms, Sunita. So Macintosh or Linux and Windows, we have tested in all those platforms. So they all of those softwares work well in all those platforms.
So you're going to walk us through how to download those in our own systems?
Yeah, definitely.
Yes, and if I need an access to a greater chunk of data in to download and get, like, see, processing is faster on the platform, right? So if I want to get access to greater chunk of data that I want to download, so that's also possible, right?
Absolutely, yes.
We will teach you to how to access the data in an efficient way.
All right.
What are the repositories that you can use to download the data or access it in other URL formats to create a more efficient data access? So we will cover that in the course of the program. Yes.
Makes sense. Thank you.
Okay. Hello. Hello. Hello.
Hello.
Yes, it's Vamshi again.
Okay.
Yeah,
So I was just seeing go through the curriculum of this course. So the data visualization is not included in that. I didn't see any data visualization. It seems like it is a separate course. Again, we have to register for. Is it that?
Data visualization is included in the program. You have seen that we will use various data visualization tools like histograms. I don't know, we'll visualize regressions, we'll visualize PCA, clustering, many other types of machine learning and data visualization. We will also customize these visualization so that they're more interpretable, more intuitive, right? So yes, we will cover those topics in the program in detail.
Okay, thank you so much.
No, thank you. Any more questions? Okay, I will repeat this for Arnab because he missed a part of the session. So we will have a large amount of exemplar projects. So we will teach you how to use data science, but we will also have exemplar projects in areas of oncology, microbiology, but also many other areas. Like we will be analyzing data types, asthma, and machine learning. Combined, we will use data science on diabetes and many others. So we will have exemplar projects in our program.
One more question from Sunita.
Okay.
So now we are having only this program, right? That is starting on September 16th.
Yes.
Right? Okay.
Yes.
So I just want to make sure I'm not missing upon anything.
No, Sunita. Yeah.
This is the only one, right? Because I miss most of them because I have my classes and I have a lab also to work in where I go. I miss most of the webinars and the Zoom sessions. So I want to make on the same page.
Yeah, sure. I mean, we will record every session and we will send it out to every participant. And then we will have these sessions. Yeah.
Yeah. And I'm also concerned about having these recordings for a longer time because they're really, really helpful.
Yeah. We can send you the videos as well.
Yeah. Like if I can have it for myself, is that possible that I have it for myself forever?
Yeah, sure. I mean, that's what I said. Just like,
all right. That's a great blessing. Thank you so much.
No problem.
Okay, any more questions?
I don't think we have more questions, but I mean, we are looking forward for you guys to join us for this journey together. Right, Darko?
Yes. thank you so much, and we're looking forward
Somebody
joining us.
about the duration of the course, duration of the class.
Can you repeat the question?
Somebody is asking about the duration of the class, Darko.
Okay, so the durations of the class will be 1 hour per class, and we will have 2 classes per week. So we will notify the ones that join the program in advance of each date for the class, but generally that's it. We'll have 2 classes per week, and we'll cover a 3-month program with 2 months of teaching, and the 3rd month will be reserved for you to start your own project, and we will be there to help you and guide you with all the issues that arise. Okay, any more questions? Thank you so much, Katya. Now, days of the week will vary. We will, you had the dates in the webinar, but I will repeat them. We will have dates like Monday and Thursday. Sometimes they will be Monday and Wednesday, but that's it. Those are generally the dates. And any more questions? Okay, thank you.
Thank you everyone for joining us here today, and we'll be looking forward for your presence on 16th of September as well. Thank you, Pratim and team. Thank you, Darko.
Okay.
Thank you,
everyone. Thank you, everyone. Thank you, Pratim. Thank you, Mohit, for a fantastic lecture. Thank you, everyone, for attending the webinar, and we look forward to seeing you in the program.
Thank you, Darko. Thank you, Mohit. Thank you all. Thank you for a great webinar.
Thank you.